we all know that any kind of living cells would be needing a carbon source, mineral nutrients. Now whole plants, they take their mineral nutrients and water from the soil, but how is it different in plant cell and tissue cultures? That generally in whole plants, vitamins and your plant growth regulators, they are synthesized at the meristematic regions or in young plants. Probably the reason is because that those are the sites where they are most needed, and then after the production, then they can be transported for various other developmental reasons to matured plants, the parts of the plant. So vitamins and plant growth regulators, as I said, are synthesized in the meristematic region. Now apart from mineral nutrients and carbon, plant growth regulators, there are two kinds. Yesterday also I mentioned. One are endogenous, which means the cells themselves produced these plant growth regulators. So there they are called as plant hormones. Now if they are synthetically produced or they are exogenously being present as signaling molecules, for example, which are then inducing the endogenous plant hormones, then they are also called as plant growth regulators. So initial attempts which were made in media formulations in plant cell and tissue culture, they included application of NOP solution. Now NOP solution is a very old media which is majorly composed of major salts. Have you heard about what kind of salts come under major class of salts? There are, have you heard of micronutrients and then major nutrients? So any examples for major nutrients? Nitrogen, carbon, phosphorus, sulfur, magnesium, as you said, sodium, calcium, and micronutrients, for example, boron and manganese, iron, copper. So NOP solution was initially, it was only composed of major salt solutions. So initial preparations of plant tissue culture media, when they were being discovered or made, then NOP solution used to be used, or whole plant extracts, or like for example coconut water we were mentioning. So coconut milk was being used, or extracts or juices from the plants or any other biological origin. So then major formulations of these synthetic media. So initially it started with non-defined media, and now we are at a stage where media is completely defined. Why do you think there is a need to come? Anyways, you see that it grows so well in complex media, living organisms, isn't it? Same was the case here. Coconut milk was being used very nicely. Juices and extracts were found to be working well. But in 1930s to 50s, there was a shift, and now we are at a stage where you can find ready-made media, but they are completely defined media. Why do you think there was a need to shift from a complex to a well-defined media? Batch to batch variation can be there. What else? Any other logical reason? Depending on how much impurities you get. But what other logical reason do you think people might have? Do you think downstream processing that time would have been the reason? What did I say in the earlier classes? That even though these complex media are found to be working well, but everything is in turn dependent on, nothing is foolproof, is dependent on so many different factors: the species, the type of explant, the genetic makeup. So depending on what culture do you want to induce, also the media composition has to be varied. So depending on what was the objective of the study, there could be a better formulation of the media. So until unless you have the clarity of the medium composition and how are these each factors affecting your objective, it will always be a hit and trial, isn't it? So gradually, depending on the purpose of the study, these different media formulations came into the picture. So starting from NOPs, then you got Miller's media, then what else? Murashig and Skoog, B5, but there has been a difference. For example, in Murashig and Skoog and between Gambor's medium, which is also called B5 medium, there is one major difference: the ratio between the ammonia to nitrate. In B5, the ratio of ammonia to nitrate is very less. Now, why this difference is important? For example, as I said, it is species specific and the kind of culture you want to induce. For example, when we were working with azadirachtin with woody plant species like neem, neem is a woody plant, and we were finding it very difficult to induce in vitro cultures, and MS media is generally used when you want to do shoot bud formation, shoot multiplication on in vitro cultures, but it was not working. So, but B5 started working. So, when we started doing, and that also I will come back to when we will talk about media optimization, when we did statistical optimization, which has the advantage of studying the interactive effects and even studying the single effects, we could find that if you remove ammonia from the medium and have only nitrate as nitrogen source, it is very useful or 100% efficiency for callus induction in azadirachtin. So, it is sometimes explant specific, species specific. So, B5 was then used rather than MS media because it has lower ammonia to nitrate ratio. Then the initial discovery also which was remarkable and which was very successful was the discovery of auxins. The first auxin which was discovered was IAA. IAA in plant is synthesized through tryptophan biosynthesis pathway. So, then synthetically also IAA subsequently was produced. Then the discovery came with cytokinins. Now, again in cytokinins also there are synthetic cytokinins and which are endogenously present, naturally present cytokinins. Now, addition also, please note that exogenous additions of these plant hormones, even if they are present endogenously or not present endogenously, they also affect the sequestration, the production and the conjugation of these endogenously present hormones or the production within the cell. So, it has to be optimized. Then came the discovery of totipotency and regulation of morphogenesis. Now, first came the callus induction, then there was a need to find a media which could induce root. Then there was a need to find, the objective became regeneration or shoot multiplication, micropropagation. So, gradually with the different objectives in mind, different media formulations came into the picture. But I must say that Murashig and Skoog medium, which came in 1962, till date is one of the most successful media formulations which are able to give results in general almost all the species. But still that does not mean it will always work. One has to optimize the composition of the media, then as well as the growth hormones. So, the rest of the factors have to be optimized. Even the environmental conditions play a role. So, these are some of the examples of different media which are now available in the market. Gambox, I said, is B5. It contains B vitamins. So, then that was another discovery when vitamin addition in the formulated media was also discovered, where three major vitamins, pyridoxine, thiamine, were added, and along with your vitamin B5. So, the nutrient media is composed of these different things. One is inorganic nutrients, then carbon sources, vitamins, growth regulators and organic supplements. Organic supplements here, any idea? Complex nitrogen sources, or like for example organic nitrogen sources. Any organic nitrogen source which is well known, well used in microbial fermentations. Constip liquor. What else? Amino acids are also added. Yeast extract, peptone, malt extract. So, coming on to the inorganic nutrients. Now, inorganic nutrients, as I said, apart from carbon, hydrogen, oxygen, there are 12 other elements. Now, what are those elements? The major elements include nitrogen, phosphorus, sulfur, calcium, potassium and magnesium. The micronutrients generally added include iron, manganese, copper, zinc, boron and molybdenum. So, now let us talk about the nitrogen which is supplied. So, in plants, in whole plants in nature, nitrogen is assimilated in plant in the form of nitrate or ammonium ions. Now, there are ammonia transporters. So, nitrate, there are many nitrate transporters. So, inorganic salts of nitrates, they are assimilated by the plant mixed in the xylem and the other through minerals. They come to the plant either in the root or in the shoot itself. It is then converted into nitrite, and then from nitrite it is converted into ammonium and assimilated in the form of amino acids and other nitrogen compounds. So, which pathway, have you heard of any pathway which is known in plants for assimilation of nitrates, where nitrate reductase is involved? What is that pathway called? GO-GAT pathway? Haven't heard. Okay. So, please go back and read. It is about how nitrogen is fixed by the plant. Nitrogen in the form of nitrate majorly. So, it is an energy driving process. It requires energy. So, active transport of nitrate happens. Sulfur is supplied as sulfates, then it is utilized where all? In protein synthesis. Inorganic sulfur may be supplied by organically bound sulfur. Organically bound sulfur is present in the form of cysteine. So, phosphorus is a major nutrient. Okay. So, then calcium, magnesium, potassium, they play a role in cell metabolism. Let us see, for example, magnesium plays a role in translation, it acts as a cofactor. Then calcium it acts as an activator and inhibitor in various enzyme activities in glycolysis. Then it is required in cell wall formation as calcium pectate. So, calcium is an important nutrient. Then calcium pumps and calcium binding proteins which are present, they demonstrate that it has a significant role in the cell wall formation as well. Now, calcium is also used as a secondary messenger. So, primary messengers are the signal molecules outside the cells, and secondary messengers, in response to the primary messengers, there is a signal cascade inside the cell which then regulates the metabolism of the cell. So, calcium is one of the secondary messengers in the cells. Then micronutrients, for example, your boron, manganese or copper or iron, they are inducers of enzyme synthesis. Now, boron is used for membrane function and it also affects the sequestration of other elements as well. So, therefore, this is also one of the crucial elements which might have come into the picture while formulating media. Now, carbon sources, it is again very species specific. As I said, generally sucrose is used. Now, coming to the question, why do you think generally sucrose is used? Ultimately, it has to be broken down to the most easily metabolizable sugar, which is glucose. So, sucrose one is, it is less costly. There are sucrose transporters present in the cell wall and also the invertase enzyme. So, it is not that all the sucrose in the media at one time initially itself gets broken down. This is a simultaneous phenomena. Sucrose is being taken in and it is also being divided into or broken down into glucose and fructose, and separately glucose and fructose are also being taken in and it is then diverted towards glycolysis, and sucrose also has, sucrose metabolism is present in the cell. So, sucrose is also taken up by the cells. So, as she said, if there is only glucose present, the growth can be faster because it is an easily metabolizable sugar, readily available to the cell, can be taken up. But one is the cost and the second is the, not all glucose is needed at one given time. So, slow assimilation is also one of the reasons. Now, vitamins for example, thiamine is essential vitamin, then vitamin B3, pyridoxine. So, as I said, your vitamins which are included in the media, they include pyridoxine, nicotinic acid, niacin, and your thiamine which is present in these media, which are called as B vitamins to induce the growth. Other vitamins which might be added depending on the species or the objective are vitamin C, D and E. So, amino acids and organic supplements, they serve as reduced nitrogen. Complex organic supplements like casein hydrolysate, then yeast extract, coconut milk, they are till date being used as complex nitrogen sources to reduce the Why are they still being preferred despite the fact we were saying that it can lead to batch variation, this and that? To see if you can control the batch to batch variation, these things can bring down the cost of the production media, because many of these are obtained as waste byproducts which are in the waste of the industries. So now let's talk about plant growth regulators. Organic compounds which affect the morphological structure and physiological process in plants in very low concentrations, they form the plant growth regulators. So as I said, these plant growth regulators can be native, can be synthetic, which means exogenously added, which can modify the plant growth. When they are native or endogenous, they are called as plant hormones, and rest are also called as growth regulators. Now as plant growth regulators, some of these signaling molecules, they are also termed as plant growth regulators. Now endogenous, which is one of the hormones which is volatile agent, is your any hormones which you know is a volatile agent and forms a part of plant signaling. Ethylene, very nice. Even these jasmonates, when they are salts of jasmonate, they are exogenously added like methyl jasmonate. Otherwise jasmonic acid is a part of the signal cascade inside the cell as well. So auxins, ethylene, abscisic acid, cytokinins and gibberellins, these are major class of plant growth regulators or hormones. Then because these are naturally present, then auxins and cytokinins, they interact and they are important for regulating growth and development of the plant. Now ethylene, abscisic acid and gibberellins, they generally form the regulatory role in plant metabolism or in the in vitro cultures. Now synthetic compounds which are not natural plant hormones, as I said, they are called as plant growth regulators, although it is mentioned here jasmonates, polyamines and salicylic acid. Now salicylic acid is also produced as a secondary metabolite in many plants, but when exogenously added, it forms a signaling part of the signal cascade agent. And also jasmonate salts, they then are termed as plant growth regulators because they are being exogenously added. Otherwise jasmonic acid is a part of endogenous signal cascade mechanism in secondary metabolism. So the response to plant growth regulators on growth and development in culture varies with culture conditions, type of explants and genotype. Now exogenously applied plant growth regulators, they affect cellular mechanism, and as I said, also the sequestration, activation, transport and sensitivity even towards the endogenous growth substance of the same kind or even of the different kind. So auxins, they strongly influence processes like cell expansion, cell wall acidification. Now it is said that auxins, they are useful in the growth or extension of the plant roots, shoots, length. So by the mechanism which is cell elongation and cell wall acidification. So now how does it happen? Auxins, they not only help in expression or controlling the expression of certain proteins in the cell wall which are called as extensins, so which loosen the cell wall, so and prepare the cell wall material, extra cell wall material. Now cell wall acidification is another mechanism where auxins are involved, in which they induce H ion concentration in the cell wall region. Because of this, what happens? Now H ion concentration has increased, the water gushes inside and causing a turgor pressure. The vacuole gets extended, forces itself against the cell wall. The cell wall has been loosened by the combined effect of via auxin, by having these expression of extensins which then loosen the cell wall, and therefore the cell wall is now extended and expanded. So this is how the cell increases in size, in length, elongates, and cell wall material is formed and the length is of the plant or that tissue is extended. Now inside the cells, the free auxins are synthesized and stored as conjugates. Now there is a purpose why auxins are stored as conjugates, so that they can be used later, otherwise free auxins can get broken down. So, in order to prevent their breakdown, they are stored as conjugates with amino acids or alcohols or sugars, thereby protecting them from the oxidative breakdown, and whenever needed, it is an enzymatic process through which these auxins are set free in the cytoplasm or in the cell. Now, IA, as I said, is a naturally occurring auxin which is synthesized from tryptophan biosynthesis pathway. Now, cytokinin induces cell division by accelerating or inducing mitosis. So, it regulates the activity of a protein which then activates mitosis in the cells. So, thereby enhancing cell division. So, therefore, if you go back and think why callus induction is an event which happens because of the combination of cytokinin and auxin, it looks logical, because there is cell elongation, cell expansion and cell division which is happening. And it is also in vitro plant tissue culture, higher concentrations of cytokinins can even lead to release of lateral bud dormancies, which means branching can happen then. So, cytokinins exert control over events leading to mitosis, and they can also, in some species they have been found that they can even promote chlorophyll biosynthesis. So, most commonly used cytokinins are zeatin, kinetin, benzyladenine. So, you will find majorly benzyladenine being used, even your thiodiazuron is also used. So, cell differentiation and organogenesis in tissue culture is controlled by interaction between cytokinins and auxins. So, now some general guidelines, but let me tell you, these general guidelines are general because for most of the species they have found to work, but still it does not mean, depending on what species you are working, you can take it as a guideline, but it is not necessary that the same concentration or the same hormone would work in that particular species. So, optimization is still essential. So, what are these general guidelines? Plant growth regulators, a combination is required for sustaining the growth of in vitro cultures. Now, when you need cell proliferation, which means cell multiplication to happen, 2,4-dichlorophenolic acid, which we know as 2,4-D, or 1-naphthalene acetic acid, which is NAA, or a cytokinin. So, these are auxins. Then cytokinins like kinetin, benzyladenine, and I said thiodiazuron. Now, for regeneration, which needs organogenesis to happen. So, for regeneration, what is in general found to work is you reduce the concentration of auxins and increase the concentrations of cytokinins. So, generally it is seen that once you reduce the concentration of auxin to cytokinin, which is a ratio of auxin and cytokinin, then it leads to organogenetic events. 2,4-D in dicots induces cell proliferation but suppresses differentiation. While in monocots in herbaceous species, it has been found that high concentrations of 2,4-D can lead to morphogenesis or organogenesis, thereby leading to even somatic embryo formations. So, how is the media prepared? The media is prepared in the form of, generally these are the recommended guidelines. You use 10x to 50x stock solutions of your major salts. Micro salts are autoclaved and separately, and including your iron, it is in the form of stock autoclaved separately, and then they are mixed at diluted concentrations depending on your media formulation. Then vitamins, because some of these vitamins are found to be heat sensitive, heat labile. So, vitamins, depending on what vitamins you are using, care should be taken whether you can afford to add them before autoclaving, or they should be added and filter sterilized after autoclaving the major and micronutrients. Now, in general, the plants grow well in acidic pH, which is nearly acidic in the sense slightly acidic pH, which is around 5.8 to 5.8 around. So, or 6, 5.8 to 6. So, the pH is the initial pH of the media is then maintained after autoclaving, because during autoclaving the pH can get disturbed. So, after autoclaving the pH should be ideally maintained to 5.8 to 6, and then gelling agents are added if you want to work with solidified medium. Acidic pH would prevent gelling. So, the concentrations in general preferred are 0.8 to 1% weight by volume of agar or any gelatinous substance.